All very good evening to everyone.
Yes.
And like every Sunday, this Sunday also we have our special guest on Sunday Talkies. Sunday Talkies is the initiation of Aqua Doctor Solution in Aqua One Center, Kolkata, funded by NFDB and managed. Like every Sunday, we try to organize different topic interested on fisheries sector, by which the fisheries sector people and also who wants to come into fisheries sector for entrepreneurship approach, they can also learn and they can also take different kind of message, and they should also understand how diverse this aquaculture and the fisheries sector is. So today among us we have also have one very renowned fisheries professionals, Dr. Sophia Priyadarshini Dash Madam, Assistant Professor, Amity University, Amity Institute of Marine Science and Technology, Amity University, Uttar Pradesh, and India. As we know, this Amity University also they are into aquaculture and fisheries sector, especially on marine science and technology aspects. So today, Madam will be addressing us on molecular different kind of molecular techniques uh, which our fishery sector need, or maybe how we can understand that molecular different kind of techniques also in coming days can help us to grow this blue revolution sector in a bigger way. So without much wasting time, I will hand over. I will like to request Madam to take these sessions on the head, and please go ahead with Madam presentations. Over to you, Madam, please.
Thank you to Aquaculture Solution, and also thank you to Mr. Diptanu Hermans, so to give me this opportunity to present my work and also to educate myself and also to the gathering to know something this gathering, what all uh, fish farmer needs for this molecular techniques, and also to know about them what all they need and what kind of techniques this fish farming is nowadays doing. Okay, so first let me tell you, like, is it visible? So, welcome to session. And myself, if I can say, I'm Sophia Priyadarshini Das, working as an assistant professor in Amity Institute of Marine Science and Technology in
Ma'am?
Amity University, Uttar Pradesh. So, okay. So, thank you, Mr. Deptano, and
Yeah,
sorry
please.
for this delay.
No, no, please go
So
ahead.
now I'm just Yes. So now I'm just telling you about this molecular techniques for a new era of fish farming. So before going to this molecular era, we must know what is this fish diversity or global fish production in India. So as you know, India is having a vast coastal area along with many riverine resources. If we can find out only for the aquaculture, we are having lot many Himalayan origin riverine system, this freshwater system. So India is home to more than 10% of the global fish diversity. So presently, the country ranks second in the world in total fish production. So as it whole, marine, brackish water, and freshwater. Okay. And with an annual fish production of about 9.6 million metric ton. So from this 9.06 million metric ton, freshwater only and freshwater aquaculture only contributes to over 95% of the total aquaculture production. So as you know, we are totally depending upon this culture purposes freshwater aquaculture system. So if we can see this aquaculture sector, aquaculture shares in India's fish production. So from 1980s, it is going it is growing. So if as you can see from my screen, share of aquaculture in 1986 is 23%, wherein share of capture fishery is 77%. As you can say capture fishery is your marine, totally marine production. So now in 2016, you can see it is growing up to 53%, and the share of the capture fishery is 47%. As we can always think of in 2030 the share of aquaculture it will grow to 62%, and as in this way capture fishery is going down. So when we are just telling about the freshwater aquaculture species and freshwater aquaculture species in India, the most common species in aquaculture, as I think many of you are having this aquaculture farms and all, so you know that there are most common like your Indian major carps, rohu, katla, mrigal. They are the mainstay of aquaculture, and they are the favored species in freshwater aquaculture system. And then we can have this pangasius, that is a catfish, tilapia, prawn, and like freshwater prawn. So these are the freshwater farming system. And then we can go for some kind brackish water species, white leg shrimp. That is a popular brackish water aquaculture species as compared to tiger shrimps. So these are the fish farming. Is a like historical, if you can say, in Mahavatar, in Kautilya's Arthasastra, can bring out fish culture. So it's a long process and it's a age-old process. Like you can say, or many of you are from West Bengal and all. So previously, we are depending upon our backyard pond for the freshwater cultures. Like when there is no such culture system or big fish farms and all, there also, as we are having some backyard ponds, we are just culturing some rohu, some species, okay, for our daily consumption. So when we are going for this freshwater fish culture or any techniques, the breakthrough in freshwater aquaculture came into existence when there is artificial propagation started. Okay, artificial insemination started, and then
তার
this
তারে কো
induced
তোমরা
breeding
অটো
started.
করে তাইতো
That
তুই গে
is the
সাবিনা দিয়া
breakthrough in freshwater
প্লেটে নাও
aquaculture. So when we are having fruit fish, we can inject them. So why we are inject them? We are inject for the growth of their eggs, and they can release those eggs. So as you know, this freshwater system, when we are having our rohu, katla, mrigal, any of these species, okay, so they can grow, they can mature there, but they cannot release their eggs. So what happened? We need to have injection. We have to give them hormonal induction to raise their release their eggs. So for that, this revolution like if you can say, this revolution came into existence when this induced breeding process starts. So why we need this induced breeding? So as I have told you, like they this species, the mrigal, or you can say any kind of catfishes, they won't breed in captivity. So as we are having, like we are culturing them in fresh water, stagnant water, ponds. Okay, so what happened? So they won't able to breed. So induced breeding is a technique where organism is stimulated by some particular hormone or synthetic factor providing opportunity to breed in captive. So this promotes release from the egg. So I know many of you maybe also are with this hormone induction and all. What are doing? They are working on the motor level. So pre-this breeding, why we need, before going for any kind of induced breeding, we must our brood fish ready to induce them. So brood fish, as you know, the brood fish, like how to select brood fish. So we must have some idea on our mind to select this brood fish. So selection of this brood fish need the time of induced spawning should be healthy and fully mature. So we need some fishes; those are healthy. So how we will know that these species are healthy? The fish must show good growth. So how we will know that good growth? How to detect good growth? So if you can say in age of old process. We are going for this classical genetics and classical way of farming. So we can see those species are going growth. Like in 5 months they will grow, till 8 months they will grow. Those we can select those, and we can feed them. Thus, this molecular level thing, or there are subjects in this. So, what is genetics and what is it? So, genetics is the science of heredity and variation. So, as you, if your mother is having some baldness, okay, so the person, the son, the eighth generation, you can say. So, the person is the son is having some tendency for this baldness. Why? Because genetics. So, when the mother father is healthy, they will give rise to a healthy individual. Okay, so if the mother father grows very spontaneously, they will grow. They will have good flesh quality. They will have very good health condition. So they will give rise to very good F1 generation or the offsprings. So there came the genetics. Where the genetics play a very important and crucial role in the of heredity and variation. So transmission from generation to generation. So used through the process of reproduction. And factors which causes the offspring to resemble their parents. So as I told you, like if we have two rohu and rohu good like you can say we can test them and we go for the breeding process so that the offspring have some hereditary character from both the parents, and they will give rise to a good individual. In this case, good means your fresh quality, because we are going for a farming. So what we need? We need flesh, good flesh, and we need a good healthy fish. That would be disease resistant. That would be mortality good. You can say growth must be there. So those things came from genetics. So when we are selecting for breeding purposes, we need the genetics. Then we need the potentials for deleterious results, like selective breeding, rationalized what? Color, size, shape, better FCR, feed conversion ratio, reproductive capacity, disease resistance, all those things needs to be there. So now this selective breeding can be done by phenotypic appearance. You can see the phenotype, you can see the outer ends, you can take them, or you can go for the molecular reading. You can see the molecule level, then you can select those species or individual. So in molecular breeding we can go for hybrid seed purity testing. So I think if you can go, if some of you are from fish farming background, you must know that if you are going for seed collection from market, okay, nowadays what happened, we are just collecting, we are purchasing seed from market. So what happened when you are going for rohu seed, and you are if you are not that much aware of how it looks, how what is the similarity pattern from bata or some reba. So a person can easily give bata seeds instead of rohu. Okay, Leo bata instead of labia rohu, rohita. Why? Because you are not that much ensure about the phenotype. Okay, the outer appearance, the morphology. So what happened? Like those two species look similar when they are in the fry or cutting stage. Okay, so what you can do? You can go for acidity testing. So hybrid testing can be done through molecular level. So you can go for a simple PCR test, maternal inheritance test through the mitochondrial DNA, and you can get pure seed. Okay, so that could help for your fish farming. DNA fingerprinting. If you can go for the paternity test, those feces are having, like, those are some feces we are just going for like breeding, and we need to know which two animal we have bred. Okay, we have bred. So you can go for DNA fingerprinting, and you can say these feces are from these parents. So that should be that can be done through molecular breeding only. Then you can go for the marker assisted backcross or marker assisted selection. So here you can ask me, what do you mean by marker assisted selection? So marker assisted selection is the selection process now we are doing through molecular technique. Okay. Previously, what we are doing in the classical genetics, we are seeing only the phenotype. But here we can do this marker assisted selection through molecular markers. Okay. So what do you mean by marker? So marker is a term. So when we are just reading something, we are marking in a pen. So that is also a marker. When we are going for some route from, you can say, from Delhi to Kolkata, we are just going through seeing these milestones. So the milestone markers, so how long we have to go, how much distance we cover. So those are also the markers. So these kind of markers, those can mark something. Okay. So markers are maybe of, like, you can say, biochemical, morphological, and molecular. So biochemical markers are isozymes, proteins, all those things. Those can be helpful for the biochemical thing. Then your morphological. So morphological markers, you can go for the classical genetics. So how it looks, how the if you can go for this ornamental fishes, then you can see the colors and all those things. Like, those are morphological markers. Then you can go for the molecular markers also. So you cannot, if that is much more difficult to understand. So molecular markers are there. Those are based on the hybridization. So RFIP, mini satellites, microsatellites, or you can just simply go for a PCR based molecular marker identification. So there are APCR, LPD, ESD, ISSR, STRs, SSRs, then your SNPs. So those are the markers that can give you a better insights for this breeding, molecular breeding. So when we are going for this molecular breeding, so we must need some genetic diversity. So as you can see, if there are no such diversity is there, so if just think, there are in the planet, there are single person is looking like same. Those are having the same gene organization. So what will happen if you go for repeated just mating of those two animal, any human or any animal? So they will give rise to nothing, the same thing. Okay, those two, just see, a is there everywhere. So if this two a will breed, they will give rise to another a only. There is no diversity. But when there will be a slow growth, a high and better growth, that is having different types of traits, okay, then you can get a new variety. So to know a new variety, there must be some genetic diversity. As you can see, we are having lots of riverine system in India. So we can get lots of different types of food for fishes. Their growth is different from, if you can see, Mahanadi, if you can go for Ganga, you can go for Kaveri. Okay, so let's say it's one is from eastern region, one is from northern region, one is from southern region. So the food availability is different also. But the species, if you see root, so species is same, but climate is different, environment is different, ecosystem is different there. So what happened? They can, some species can get good amount of food. They can grow much good way. Some are having not that much good growth. So there are some diversity within same species. Okay, so along with, you just, there are different river and system. If there is only one river system, but the river also is having a long stretch. So just think a river Subarnakha is there. So it is having a long stretch. So there are different area, and you can get good food or the appropriate food for those species in some area, and some area doesn't have anything. Some area is polluted. So what happened? There is also genetic diversity you can find in between those areas. So augment genetic diversity in a fish species is fundamental to improvement. So when you go for any kind of improvement of fishes, okay, not only fishes, any kind of animal, you must need genetic diversity in that particular species, that particular, like if you can go for that particular species. So genetic variability is considered the reservoir that fish breeders fall upon in their continuous strives to develop improved varieties and hybrids. So to know, like the knowledge of germplasm diversity and relationship among alike breeding material has a significant impact on improvement of the fish. So this information is useful in planning crosses for hybrids and line development and assigning lines to heterotrophic groups. So as I have told you, I am not going for the core molecular markers. You can see the features or properties of different molecular markers. So if you can go for the RFLP, restriction fragment length polymorphism, that can be detected through hybridization. You can have the gDNAs, like from the fish you can get a small amount of the fin and it can regenerate. This is no harm you are giving to the fish. You're just taking the genomic DNA or the tissue, and then you can isolate the genomic DNA. It require a radioactive activity. Then you can go like in this RFLP. It's a very age-old process, and the genomic coverage is limited, and you can get a low degree of polymorphism or variation. So phenotype expression is codominant. Codominant means it's from only, it's a two-way. Then possibility of automation is no. But if you are going for nowadays, what we are thinking for this molecular breeding, microsatellites and SNPs. So microsatellites are the method we can detect through PCR, and you can have the sequence specific primers. Sometimes it will require radioactive activity and some, but extensively genomic coverage you can get, and polymorphism you can get more. So if there is more polymorphism, you can get better traits. There is the phenotypic expression is dominant and automation is there. But nowadays, above microsatellite also we are thinking of SNPs, single nucleotide polymorphism in our fish DNA. So that SNP can be detected through PCR. Okay, and you have to just go for the sequence specific primers, and there may be requirement of radioactivity or not, but the extent genomic coverage is extensive, and you can get polymorphism high because it is a single nucleotide differentiation. Or sometimes it is deletion, insertion, many things will be there. Codominant expression you can get. So these are some properties of different molecular markers. So what are the application of these molecular markers? I have told you in the beginning. Fingerprinting, DNA fingerprinting, diversity studies. We can have this diversity studies, marker assisted selection. We can go for the genetic maps, gene tagging, novel allele detection, based gene cloning, F1 identification, comparative apps, bulk segregated analysis, and seed testing. So fingerprinting, for this paternity testing, diversity studies for better variant, better trait. Then we can go for the selection, marker assisted selection for a better, like it's a holistic approach. Then came your genetic maps. What does genetic map know? Genetic map give you all the information about your genome, about your chromosome, as you know, different fishes have different chromosome number, and these chromosomes are having some gene, and those genes are responsible for growth, responsible for color, responsible any kind of thing that can you are just seeing. I'm just talking about some work of some gene. Okay, so all those things are there in our genetic material. So those genes, you can see those chromosomes, like these are LG 13, 14, 15. It's from one of my work. You can see this LG 13 means linkage group. So linkage group and chromosome are somehow we can assume these linkage groups more or less similar to the chromosome. Okay, and you can see there are some points are there like FHE 903, FHE 2156. Those are some locus. Okay, so these locus are taken by some markers, molecular markers, and that can be driven by some kind of gene. And just think, just to know this LG14, you can see there is one BO8 is there. So just think this BO8 is responsible for growth. So what happened? This BO8, if we can isolate from one organism and we can just insert in another slow growing fish, so what happened, that slow growing fish also get enhancement. But as I can say, these are not so easy. Okay, but this kind of gene insertion, knocked out, all those things are called genetic engineering, and these can be done. Okay, so and also if some growth is there, the growth is not only responsible by only one gene, it is the effect of many genes. Okay, so these kind of things you can do through molecular approach. Then came mass. What do you mean by mass? Mass existed since evolution. Think there are different substance. Okay, so for a you can say classical genetics, I see a growth phase. You just take some feces and you just see this is much more different in, and your knowledge is there. You cannot see without seeing without looking on the other appearance. You just this is substance, you are sure that this is registered. Okay, before going for selection, before going for so for that reason, there will be you can go for disease registry easily. You can see from the you have to nitrogen population analysis. You have to select various species, various animal from different area of your data. You can get many variety of species, and you can go for see, if you are having fish that is more or less disease prone, like kind of tuberculosis there, so then you can put her to organisms. Okay, so just see, that is called a pathogen challenge or disease challenge test. Challenge give, you can give them pathogen, you can see how they are surviving. Okay, so some time if you just those species are there resistant to that pathogen, those species you can take and go for genome sequencing. What we will do, what you get from that genome sequencing, you can get some diseases which are registered, those are into what genetics part. So genetics, if you can see, it is because we have to select a brood. So why we need this genetics? Because this is the science of heredity and variation. So heredity transmission from generation to generation, usually through the process of reproduction. And you can find out healthy mother and father will give healthy offspring. So for that reason only we can take the help of genetic variation. Okay. So next.
Yeah.
So from this genetics only, we need to go for selective breeding. So to know the father, mother, the size, color, shape, better FCR, so we can get into the selective breeding process so that reproductive capacity, disease resistance, all those things are taken into consideration. So before going for the selective breeding, next, sir,
Yeah.
we can go into the molecular breeding. So molecular breeding is nothing but taking the molecular techniques into consideration for hybrid seed purity testing, DNA fingerprinting, marker assisted selection and heterotic grouping. So hybrid seed purity testing. So as you maybe know, like there are, if you are going for collecting some seeds from the market, you can get lots of mixing of all those different kind of seeds. So now you can find out the pure seeds, then hybrid seeds. If you can go for like Rohu Katla hybrid, there are Rohu Mrigal hybrid. So you can test those hybrid seeds through molecular techniques. You can go for the paternity testing through DNA fingerprinting. You can get good amount of selection through marker analysis. So next, so what is a marker? Next to next.
Yeah.
Okay. Hello. The next. So markers are some benchmarks. You can say if you are going from one place to another, you can see some milestones. They are also markers. Okay, markers are those can mark something. So markers may be biochemical, morphological, or molecular. So biochemicals are isozymes, proteins, and morphological are those are like you can see. Okay, the morphometrics, the morphology. Okay, then came your molecular markers. Those are based on the hybridization like RFLP, mini satellites, microsatellites, and some are based on PCR. So like RAPD, EST, ISSR, SSR, AFLP, SNP. So next, so to get these markers, we must know some genetic diversity. So genetic diversity came into existence when we have variation in the gene pool, and this variation can lead to improvement. So what type of improvement is there? The trait improvement. What type of trait we want to consider? Just like there is growth is a trait. We want to improve our fish growth, so that is called improvement. If there is a color enhancement, we can go for the improvement of the color, so that is also through genetic diversity. So assessment of genetic diversity in a fish species is fundamental to its improvement. So we must go for genetic variability and is considered the reservoir that fish breeders fall upon in their continuous strives to develop improved varieties and hybrids. So the knowledge of germplasm diversity and relationship among the allied breeding materials has significant impact on improvement of fish. So why we are going for all these techniques? Only for the improvement of fishes, to get all those improved quality of your fishes. Okay, so next. So genetic diversity also gives you some features of your molecular markers. So everything is depend upon your molecular markers and molecular techniques. So as you can see, RFLP is a low type of polymorphism detection type of marker, whereas microsatellites and SNPs, you can see they can detect a high quality of polymorphism, so that you can get a new variety of fish as early as possible. So next, so DNA marker application. Next, sir. So DNA marker application. So application of this DNA markers are in different ways. I can tell you in the fingerprinting, DNA fingerprinting, diversity studies, marker assisted selection, genetic maps, gene tagging, novel allele detection. Okay, all those things are there. So next, so before going to this marker assisted selection, we must know genetic maps. So what are genetic maps? So how we need this in fish farming? So as you can see in this picture, some LGs are there. So these LGs are linkage group. Linkage groups are nothing but your chromosomes. Okay, so similarity to the chromosome. If you can see, LG14 is not the chromosome 14, but it may be chromosome 14. Okay, so you can see there are some markers there. So these marks are your markers, and these markers are driven by any kind of gene. So just let's say one gene is there in LG14. Okay, so what happened? And this gene, this marker is responsible for high growth. So you can isolate this type of marker, and you can, like, you can insert this marker to another slow growing fish, so that you can have a fast growth of your fishes. Okay, in map can give you lots of information, not only the growth, but also there are many types of application to this genetic maps. Then came next, your molecular assisted selection. So when you are just seeing, if you can go for showing some molecular assisted selection, sorry, if you can go for selection, so you can tell me, we have will see one fish is higher growth and a lower growth, so we can only take the higher growth fishes. But when a fish is prone to disease, so how we will segregate between disease fish and non-disease fish? Like those can be in a in a way to get disease. So for this reason, you must need some molecular assisted selection. So here what happened, you have to take more and more population from the gene pool, and then you can give them a pathogen challenge, so that those species are alive till the end. Okay, those are disease resistant fishes, and from those disease resistant fishes, you can go for a genome sequencing. You can have the genes, those are responsible for disease resistance. Then you can go in the in silico process for the functional annotation, gene expression, comparative genomic study. Then what happened? Those functional genes or candidate genes you can evaluate, okay, in vitro, and then you can target those genes to a different animal, and that different animal will, like, different individual, and that different individual will give rise to a like resistant animal, and then you can just go for breeding those resistant animals, so that you can have a resistant gene pool, you have a resistant population. So this is the marker assisted selection, where you can get this disease resistant feces. Then came your genetic
Yeah.
engineering. So genetic engineering is nothing but, is a, like, including chromosome sets and manipulating transgenesis. So genetic engineering is engineering of any kind of traits. You can see these glowfishes. These glowfishes are manipulation of colors. This chromosome set manipulation of modification, like when you modify something. Okay, so what you are modifying? You are modifying the color. You are just enhancing the color. So normal organism have two sets of chromosomes, and one from each parent. So one type of chromosome, when, like, manipulation we can do, then you can get a better version. Okay, so these animals are generally sterile, but okay, but can be advantageous under condition when reproduction is undesirable. So when you can see some feces are there, non-indigenous weed controlling grass carps to prevent unwanted reproduction. So those things you can go for chromosome manipulation, or you can go for genetic engineering. So next you can see a video of glowfish where just click it in between. Yes, just click on the middle of that. Okay, if it is not working, then we can go to next. So glowfish also is a type of like genetic engineered fishes. You can see from that picture, like there are different types of color you can find out, and those are called your green fluorescent proteins. So now you can see in the picture, those are your transgenic fish. So transgenesis is also another type of genetic engineering. Okay, it refers to the technique of incorporating a gene or bunch of genes through biotechnological method, or you can say molecular method. And you can go for, like, you can see one fish is growing so high and one other is growing, it's so slow. So why? So this is a salmon. So why these are showing so much of differentiation? So the first one you can say is your you can go to the next slide. It will be better. So you can see these are your aqua advantage salmon. Okay, what do you mean by aqua advantage salmon? So aqua advantage salmon is an genetically engineered Atlantic salmon developed by Aqua Bounty Technology in 1989, and the typical growth hormone, that is regulating gene in Atlantic salmon, was replaced with the growth hormone regulating gene from Pacific Chinook salmon. You can see here. Okay, so what happened with the promoter sequence from ocean pout? So the gene enables the genetically modified salmon to grow year round instead of only during spring and summer. So what happened? You can get a good amount of growth. So what happened here? These genetically modified fish are able to grow even faster and grow to market size in just 16 to 18 months rather than 3 years. So what happened? You are just squeezing the time. You will be getting a good growth and faster growth of salmon in about 16 to 18 months. So whereas in a only you can go if you can go for a normal salmon, it will take you around 3 years. Okay, so these are the advantage of your salmon, sorry, genetically improved salmon. So genetically engineered fishes. So next is your gift. Gift is your genetically improved farm tilapia. Okay, so we have seen genetically modified and genetically improved. So genetically modified uh, terms will be there when you are just putting some amount of genes there. You are just modifying something. But here in gift tilapia, you can see this is a genetically improved farm tilapia. What happened? This is the important candidate species, and here in tilapia you can see those are modified or improved by selective breeding. Okay, so in this genetically modified farm tilapia, it is successfully produced a farm, and you can see there they are reaching around 600 to 900 gram for from 50 to 80 gram in size in 6 months. So this gift tilapia gives us a fast growth, disease resistance, ease of farming, low production cost, high survival rate, high stocking density, farmed year round, and you can find out tolerant to lower oxygen level, tolerant to lower water quality also. Then came next, then came another genetically improved fish, and it is also a farm fish. It is the Jayanti Rohu or genetically improved Rohu. Okay, so selective breeding program in ICR CIFA. Okay, it will gives us this kind of rohu. So whatever you have seen from now, above this rohu, it's from the other country. But in India, we have also having a genetically improved fish, and it is showing about 18% of genetic gain. Okay, and how they are doing it from gene variation. So they have selected many species from different riverine system, and they can just go for the variation, and this variation leads to genetic improvement. So this is also a molecular technique. So next, so there is also nutritional biotechnology in molecular way. You can see in the feces we can have alternative protein sources. We can have flesh pigments. We have better PUFA. We have omega-3 fatty acids in the profile of fish flesh. So it can also give a molecular level, like we can go for this farming. So next, so why? So these are all new era of molecular technology. So next, so now coming to this new era. So till this nutritional biotechnology, what we have seen, those are giving us some kind of, like we are just going through
স্যার আমার একটা
selection,
ক্লাস
we are
চলছে
just
শেষ করে
improving
আপনাকে
through
কল করব
some molecular technique. But when it come to whole genome sequencing, so nowadays whole genome sequencing of feces or any kind of farm animal, any kind of livestock animal gives us more insights into these disease resistance, this growth, any kind of traits that you can find out. So whole genome sequencing means you can have your genome profile, your fish genome profile, and whatever lacuna you are getting in your fish, you can change it, you can modify it through any kind of molecular technology, any kind of molecular engineering. So this is the new era of molecular technology where you can get lots of benefits to the farmers. Next, so if you can see this Labirohita, you can get genetics, genomics, epigenetics, metagenomics, transgenics, genome editing, and transcriptomics. So genetics, it includes more traits, dissemination to global level, automated phenotypic, and sterile rohu production you can get through genetics. When it comes to genomics, you can find out quantitative trait loci, identification, marker assisted selection, development of single nucleotide polymorphic array, and genome-wide association study for genomic selection. So you can get tissue-specific miRNAs, methylation, interaction, development of epimarkers, and you can go for metagenomics like identification of microbes, good microbes, bad microbes, linked with health, nutrition, and reproduction of feces, and also you can go for probiotic development, antimicrobial resistance. Then you can go for transgenic and genome editing, where you can go for CRISPR-Cas9. You can go for development of optotransgenic rohu. You can go for transcriptomics. You can go for tissue-specific gene discovery, differential expression of genes. So all those things you can do in this new era to help farmers. So the benefits. Next, so benefits of this molecular markers or molecular technique will be for culture of fast growing aquatic animals, and it increases following, like this protect water quality, also marine sediments and aquatic health, marine and as well as freshwater, and it reduces cultivation time, and it could supply, and it can be increased without proportionately increasing the use of coastal water. So if you can go for some kind of shrimps, brackish water, so you cannot go for only, you cannot rely on only coastal waters. You can go for any kind of like genetic modification, and it can grow in any water. So higher food conversion ratio you can find natural resources, you can require like many to produce them, and sustainability of fish farming. Okay, so it also improves the economics of aquaculture, so that land-based system becomes economically viable and competitive with ocean pen system. So we can also competitive to ocean. Okay, So in this way, I can say molecular technology can give you a better insights into farming system, and you can have a better farming technology if you can just apply molecular technology to the farm pieces. So next, and thank you for patient hearing, and thank you for being there with lots of glitches, and hope you'll be having a good time with this farming techniques.
uh, Thank you so much, ma'am, for your very interesting topic you have addressed. Though we know many people that in fisheries, genetic engineering and biotechnological tools are very important in 21st century, because whenever we stock fish, we always want my fish should grow fast. So to make
Yes.
my fish to grow fast, we need lots of biological tools, or say the, best quality seed, best quality brood stock, and also the genetical engineering part little bit, so that our fish should grow fast compared to other natural environment fishes what we are having now. So in that aspect, you have addressed some important parts, like you put the example of gift tilapia. You also address the genetical engineering part on salmon. You also address out the jayanti rehu, and also now many people might be heard about amur carp, which also a process of genetically modified uh, group of fish people are now trying in the culture system. So thank you so much once again for your nice presentations, with very simple explanations, with simple pictorial presentations. You have addresses out by which I think most of the people who join last and also join before, they have understand, though in between we have some technical issues on internet, but later on it's solved out, and also nicely you have ended the sessions. So in the meantime, if anybody want to ask any questions, session is open. You can ask the questions to Dr. Sophia, madam is here, so can address the questions directly from your side. If anybody want to ask any questions, please unmute yourself and can ask the question, please. Anybody want to ask any questions
I
to?
think either I am totally they have understood everything or so okay. Someone asked me what is
Yes.
RFLP. It's a randomly fragment length polymorphism. Okay, random fragment length polymorphism, where we can have some polymorphic DNA, and you can find out how this variation is there. So as this RFLP technique is so age old process, it's obsolete now. So you can get diversified or variation from a fish. So if you are having a group of fishes, or if you are having a you can say population, so from the population, if you
অমিত কে
want
বল
to
না একটু
get
বাইরে গিয়ে
some
কথা বলতে। অমিতকে
variation
বল বাইরে গিয়ে কথা বলতে।
sample, okay, then you need to have this RFLP technique. But now we are having SSRs and SNPs. You can better go to those markers, so that it will resolve all your problem. Okay, I think I may give you answer, Mr. Narendra. Oh,
Okay,
thank
okay.
you.
Thank you. If anybody want to ask any more
No,
questions, yes, sir, please. Azad sir.
the presentation. I don't. I was thinking that maybe my net is problematic. No, there was some break in the audios. Anyway, it was solved out. Madam, this you are targeting a gene for improvement, like say for example growth.
Yes.
Is there a chance for suppressing another factor?
Yeah. If you can see, sir, if we are going for any kind of growth, if we are just thinking of a growth gene, so it may affect. I cannot tell you that it may not affect, but growth is not a single, like there are some genes and some traits that are not only travel by only one gene. It could be a multiple gene oriented traits. So if you are just enhancing growth, so there is, you must say leptin. Leptin is for growth enhancement, muscle enhancement gene. So if you can go for muscle enhancement, so it won't give you that much difficulty or that much, you can say, negative impact. But if you can use it if it is growing and growing in a bigger way, so you can get some deformities that is there. But if you can just give only one gene, if you are just targeting one gene only, so it won't give you any kind of miss, you can say, bad impact, I can say. But as you say, there are some negative and positive impact always. But if you can go for this, you can say gene targeting, I can say there will be, till now there is no such kind of you can say reports are there. So you can see those glowfishes. There is no issue at all when you are targeting a simple gene to insert in this fishes. When you are just thinking this cold tolerance gene in salmon, there is also no issues at all. So there is no report till now. Like there is some issue with this genetic engineering. So I think
Yeah,
I'm able to.
yeah,
Yes.
yeah, of course. That why I said is, you have different traits which you target in
Yes.
genetic engineering, right?
Yes.
So by enhancing one, maybe another which may contribute to a greater extent could get suppressed. Like for example, growth, and you are getting say 10% growth, for example,
Yes.
by manipulation. And if you are suppressing the resistance factor or the immunity factor,
Mhm.
like diseases may contribute to 20-25% or sometimes 100%.
Yes.
So in that case, you will have more losses. Though you are targeting at 15%, you will get more losses. So is there any chance of that kind of this one?
Yes,
Are there
sir.
any instances or
Sir,
examples?
in this way, you can target, you cannot target only one gene. You always target multiple genes so that your, when you are just enhancing something, it can't suppress the other. Okay. So if you are just going for a whole genome sequencing, so you are not targeting only the disease resistant part or only targeting the growth genes. You can target both of them, as I have shown you in the picture, the genetic traits, the chromosomes. You are not targeting only one, because if you are going for a molecular enhancement, so you cannot just look for only one. And before going for targeting some gene, so you must ensure that will not give you any bad impact. That must be needed. Okay, sir.
Uh, yeah. No, why I ask this,
Yes.
lot of people who are listening to your talks, most of them are, farmers, and then, those who are practicing in the field, may not be professional. You know,
Yes, sir.
they should not misventure into these type of techniques. Now these are all the techniques which have to be studied very in a complex way, and
Yes,
then
sir.
like what you said,
Definitely.
you have to see what is the effect of one on the other. So all those things have to be seen. So those who are in the field, they should not simply think that okay, I will increase this and I will
See,
increase that.
sir, let
That's
me just add something here, sir. So this kind
yeah,
of technique cannot be improve in overnight process and in a farm
yes,
condition.
you
You
are right.
have to develop this thing in a proper molecular technique, in a proper molecular biology lab, and then when you get the positive response, then only you can disseminate this to farmers. Not when you don't have this particular thing that this will not like giving you any negative impact, then only you can give it to farmers. So farmer, if one farmer will be there and he want to enhance the productivity through gene silencing or enhancing some gene editing some gene, so it can't be like he can't able to do that. So when someone like gift tilapia, everyone is now using gift tilapia. How? Because someone has raised that thing and someone done like does that thing. So then it will market it. Okay, then it will come to the picture after it is done. So in this way, when there will be some growth enhancement, this growth enhancement can be done through a molecular biologist and a group of people. Then it will be just marketed like, you can say now coronavirus is there. So everyone knows that coronavirus is there and there is having some issue and they are just going for the vaccination. Okay, so till the vaccination doesn't go for the trial, it will not go, like it will not approve, you are not applying it to human. So in this way, how we are just thinking about human, we are also fish farmer or fish scientist, we can think about fishes. So whether the technique is not approved by a whole lot of bunch of scientists and it is not approved from the higher authority, you cannot get into like give it to any farmer. So farmer never think like in one day or overnight we will just produce a 1 kg 500-gram fish to 1 kg fish. It is not at all possible. Okay, but there are some possibilities like this gift tilapia. It's showing so much of high growth because you can have this strain, gift strain. There is a normal tilapia is also there, Oreochromis niloticus, but there is a strain of Oreochromis niloticus that is a gift strain. So that gift strain is only giving you this kind of high growth rate. Okay. So, sir, is it clear now? So, I think
Yeah, yeah, thank
if
you. I
any,
know.
yeah,
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 I
no, thank
think
you. I know that knows what I am talking about.
yes, yes,
I
yes,
know.
yes, indeed. I think I will just make it a bit more simplified to all the audience. Good quality growth is leads to good quality offspring. Good quality offspring will lead to good quality production. So that's what sir and madam was trying to address, that genetics part is always important, because somebody also tried to mention that it is a very complex method. Only just maintaining good food, good water quality will not give you good fish growth. Let me tell you, because first you need seed, then come feed, then come water, then come management, then come production. So for
Yeah,
seed, we need to understand the baby fish from which mother parents come and what is their genetic resources or genetic pool. Are they are quite good enough to give me short period of time good production? That is very important. Though in farmer level we can't do lots of research on growth promotion, but our research institutes, they are definitely devoted for such kind of research activities, by which we can also sometime listen that now we have gift tilapia generations or monosex tilapia generation F11, F9, F10. These all are the generation upgradations with period of time. Even in Amur Cup, Jayanti Rahu, there are also genetic upgradations year wise. So it's a process of good offspring lead, which will lead good production in coming days. I think I try to make the statement very clear to everyone, so that we understand that this is not a very complex method. This is a process of selections to end the offspring. So this is basically a methodologically process which ends with good quality seed productions. That's what also madam and sir was trying to address. Thank you so much. If anybody want to ask any questions, can ask. We'll take the last question of the today's session. If not, then I will ask the last question to madam. How, say, a farmer point of view, say I have, say, a multiple group of species in my pond, but out of this I have stocked different fish species from different hatchery, but though they all are rohu, but out of the rohu also some rohu shows very fast growth, some rohu shows very slow growth. In that perspective, as a farmer's uh, point of view, what will suggest to make them separate and grow them separately, which are growing big, or make them, proper feeding management, or maybe proper kind of nourishment, so that we can bring them together to be a good growth at the last? Madam, please.
Okay. So you have answered the question previously. Actually, this question answered by from the previous question. So as you know, these are from different genetic pool. That's why they are giving you, because you are not giving different food, different water quality, different environment. Same conditions are there in every other fish you are giving, but they are showing difference in their growth pattern. Why? Because of their mother and father, their parents. So there is no requirement of giving them other, it is a tedious process, and also you will be giving you can have more number of ponds or tanks. You need that. That is a cost effective thing. So you must, like, as a farmer point of view, you must cut short your price for culturing or to grow out those fishes. So you need to be there and to grow them as such. But yes, care should be taken for those lesser growth species. But yes, you can if you want, then you can segregate them and you can give them a good protein diet, so that more number of protein must be there for those species. And in this way you can grow them. So as I know, I can tell you from the molecular perspective, but you can know better to handle those species. And I can tell you, fishermen and fish farmers, they know much better than a scientist how to raise a fish. So there is no issue at all. Only the genetic part is there, so that is showing their morphological pattern here, the genotypes, so that you can get those slow growth and high growth. And always you can find out this slow growth and high growth if you are going for this two high growth fish to mate. And those feces, those offsprings, also you can find out some feces are growing so high and some are less, because that is the tendency of a bell curve. You can find out in any growth pattern. So there is no issue. You can have it in one pond and you can take a better care of those pieces. That
Yeah,
is
I
from my
think,
side.
yeah, I think I'll just ask the last questions. Last question is
Yes.
that many people, like many feed companies, many feed supplements company, they say that, okay, you keep on add this high protein and feed supplement continuous, it will act as a something, growth booster, growth hormone or something kind of structure, that your fish will have better growth. Does it really work on the ground? From your research perspective, like to know, ma'am.
See, if we are thinking of a research perspective and we can rely on that feed, and we know that that feed has those growth parameters, then yes, definitely we can rely it. But prior to that, prior to rely that fish, you have to see those feed and you have to analyze those feed, that yes, whatever parameter is written in that content page, or in that you can say box or something, so is there the same amount of things are there in the feed? So was it only for the for marketing? So that should be known from a person. So you have to just, and there are, if you can say, there are some branded feed companies are there. You
Hmm.
cannot go and you can just tell that no, 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 these are not the same thing. Yes, sometimes they are adding some growth promoters, some growth enhancement is there, but you can say this is all about the body type, all about the genes. So if it is a slow growth fish, you can just boost it with some booster package or anything. What they will do, they will enhance in 1%
Yep.
2%, as they like enhance in a better way. No. So you have to think about like from their perspective. Yes, we are giving the like booster thing to our fishes, but fish needs to be healthy. As you are always saying, healthy fish gives you healthy, you can say, offspring, and healthy offspring will give you and make you a healthy farmer.
Yeah, yeah, true.
So that is the thing there. So yeah, you can give the food, those feeds to your fishes. So everything is depending upon your analysis first.
Okay. Thank you so much, ma'am, for your nice presentation, and also it was very quite interactive sessions. And thanks so also to the participants who joined in between, also who left in between due to technical issues. So these whole sessions also will be available in our YouTube channel, that is Aqua Doctor Solutions. All the Sunday Talkies sessions are available in our YouTube channel. You please go visit our YouTube channel and like the page so that you can get keep on updates on our different aspects of the different sessions. Thanks once again, Dr. Sophia Priyadarshini Das, madam, for such a beautiful presentation. And also, I like to thank sir.
Yeah, yeah. Hello, Depton. Sorry, sorry
Sir,
to interrupt. Huh? I think you have the consultancy services and you visit farms and all those
yes,
things, right?
yes, yes,
What I suggest is, if you think that one particular farm or hatchery which is producing feed is having a record of slow growth or something,
yes.
on one side you can recommend, you can ask the farmers to maybe cut down that particular this one, or ask the hatchery people to how to enhance their the performance of the hatchlings and things like that. Now
Okay.
that way probably you have a better role in the farming sector. That's what
Yes,
I think.
yes. Very good
It's
suggestion, sir.
natural that yeah, it's
We are
natural that
yeah, we are
some growth will be slow.
yes, yes. We
Yeah.
have started some projects now. Is now last 6 months. The projects are now ongoing in in seasonal changes. We like to see the how the fish reacts and the growth performance changes. So that's the last 6 months we are working on the summer period. So now this winter is approaching, so we'll be trying with the same kind of products in different kind of modulation pattern to see the how the fish reacts. We are working now on carps, and also we are working with catfish singi. So we hope in coming days I will be also able to answer you this particular question what you have asked, sir. So now we have bring down the
There,
hatchery and also the grower both
yes.
together. So that work is going on, sir.
That's a good contribution from your
Yeah,
side to the
yeah,
farming sector.
work
That's
is
good.
on the way. So I hope in some scientific outputs also I'll be able to come up. And we are now doing a trial with a Spain based company product. So let's see how this product also act on Indian continent zone, how it also reacts on our Indian fish and Indian environment too. So thanks once again, sir, for your inputs. And I also like to thanks Dr. Azad sir to join today's sessions, and also his critical analysis and comments on the particular today's session on molecular techniques, how we can bring new era in fisheries sector. So thanks once again to Dr. Sophia Priyadarshini Das madam for such a beautiful presentations, and also Dr. Azad sir to join the session, and also all the participants who have participated today from different parts of the country and outside of the country also. Thanks once again to everyone, and hope to connect you again in coming Sunday with someone special on our field of aquaculture, on addressing on some different other parts, other topics on aquaculture in this Sunday Talkies session. Till then, take care yourself, stay blessed, stay safe, and hope to see you in next Sunday Talkies. Thank you once again. Bye.
Thank you.
Thank you, Aqua Doctor.
Thank you.
Thank you, sir. Thank
Thank
you.
you.
Thank you.
Thank you, everyone. Thank you.
See you.